So dear students, so today our topic is beta oxidation. That is the metabolism of fatty acids to yield a ATP or energy. So already you know, so our body around more than 85% or around 85% are the source of ATP, or our body can got the energy due to metabolism of the fatty acid. So remaining that was came from the protein metabolisms, so as well as the glucose metabolisms, but around 85% that was depending upon the fatty acids. Okay, so that's why, so the beta oxidation, it is one of the pathway, so that was helpful for the metabolism of fatty acids and it to yield a energy or ATP. So that ATP is required for the various metabolic reactions. So from this image you should be note down there is two structure, that one is the keratin and as well as another one is the mitochondria. Okay, so these fatty acids, it was mainly present in the cytosols. Okay, and but the beta oxidation, so that was occurs in the matrix of the mitochondria or inside the mitochondria, but these fatty acid do not cross the or do not enter the, that is the mitochondrial matrix. So that's why, so this is the carnitine, so that was helpful for the entry of the fatty acids into the mitochondrial matrix, that was helpful for the metabolism of fatty acids to yield ATP. So that was helpful for the various biological reactions. So as you know, so various beta oxidation processes are there. There is alpha beta oxidations as well as the omega oxidations. So regarding the beta oxidation, it was occurs in the mitochondria and as well as it was occurs in the peroxisms, but majorly it was occurs in the matrix of the mitochondria, and as well as the alpha oxidation, it was mainly occurs in the peroxisomes. And regarding the omega oxidation, so it was occurs in the endoplasmic reticulum. So like that, so there are three beta oxidations are there, that oxidation processes are there regarding the fatty acids, alpha, beta as well as the omega. So now we go for introductions. So regarding definition of fatty acid oxidation, the fatty acid oxidation is mostly oxidized by a beta oxidations among alpha omega oxidation. So beta oxidation are prominent. So regarding for the oxidations of the fatty acids, and beta oxidations means the oxidations of the fatty acids on a beta carbon atom. So that is the bond between carbon atom 2 and carbon. So that is nothing but the beta oxidation may be defined as the oxidations of the fatty acids on a beta carbon atoms or carbonyl group, that is the bond between C2 and C3. Okay, and at the end of the result in each cycle, that is the removal of the two carbon atoms, that is the in the form of the acetyl CoA. Okay, so dear students, the fatty acids, it consists of the 18 carbon atoms. It consists of 80 carbon atoms, and each cycle or each beta oxidation, so two carbon fragments, so they are removed in the form of the acetyl CoA. So then the acetyl CoA, it was enter in the TCA cycle. Once again it can yield the more ATP. So like that, the beta oxidations, it was the one of the important pathway, it was yield the lot of the ATP. So the beta oxidations of fatty acids, so that was mainly divided into three stages. One is the stage 1, that is the activations of fatty acids occurring in the cytosols. Without activations of fatty acids, so the beta oxidation do not begins. So that activation, it was occurs in the cytosol. So then only, so that activated fatty acids, it can transported into the mitochondria. So that is the stage 2. And this activated fatty acid, it do not directly, it can allow the enter into mitochondria. So with the help of the carrier proteins like a carnitine, so that carnitine, it acts as the carrier protein. So that was helpful for the transports of the fatty acids into the mitochondria. So like that, the carnitine, it is one of the important carrier protein. So that was helpful for transports of fatty acids into mitochondria. And this stage is also known as the carnitine settles. Okay, then third stage is the proper beta oxidations. That is the metabolisms of the, that is the fatty acids, that was occurs in the stage 3. That's why this stage 3 is known as the beta oxidation proper in the mitochondrial matrix. So that's why the beta oxidation, it was mainly occurs in the mitochondrial matrix, and also it was occurs in the paroxysms along with the alpha oxidations. And regarding the omega oxidation, it was occurs in the endoplasmic reticulums. So fatty acid, here the note is there. So fatty acids that was oxidized mostly in the all the tissues of the body. Okay, all the tissues. So their ATP source is mainly depending upon the oxidations of the ATP after the glucose. Okay, in the short period, the storage of glucose is also empty, but remaining time is continuously the ATP is required for the muscles and ATP is required for tissues. So that's why the most of the tissues in the body, so that was mainly depending upon the fatty acid oxidations. However, that is the brain, erythrocytes, adrenal medulla, so they do not utilize the fatty acids for energy requirement. So brain, it is mainly, it was mainly depending upon the glucose metabolisms, and erythrocytes, it was mainly depending upon the lactate. So like that, so these are the three sites, so where they do not depending upon the energy, so that can came from the fatty acid oxidations. So next one, regarding, we can discuss regarding the how the fatty acids, so they are enter into a beta oxidation. So dear students, so in our body, the fat, it was mainly stored as a triglyceride. So in most of the eukaryotic cells, the fat, it was stored in the form of the triglycerol. And triglycerol is the molecule. It can consist of the fatty acids and glycerol. So glycerol, it can hold the, it can have the bond along with the fatty acids and stored as a triglyceride in the many tissues. So before start of the beta oxidation, so there is need to hydrolysis of the triglyceride, and there is need to release of the or suppressions of glycerol from the fatty acids. There is need to availability of the free form of the fatty acid. So that was occurs in the here in the cytosol. So with the help of the enzyme, so that enzyme was known as the lipase. So this lipase enzyme, it was responsible for to cause of the hydrolysis of the triglyceride, and triglyceride is the storage molecule for the fatty acids. Our fatty acid, the excess of fatty acid, they are stored in the cells as a triglyceride, and triglyceride nothing but glycerol plus fatty acid. It consists of one molecule of glycerol and three molecule of the fatty acids, and each fatty acid molecule, it can consist of the 18 carbon atom, and each beta oxidation, two carbon atoms are cleaved or removed from the fatty acid. So like that, the fatty acids oxidation, So that was occurs are completed in the 8th cycle, because in each cycle the two carbon atoms are removed in the form of the acetyl CoA. So that's why if the fatty acids consist of 18 carbon atom means the beta oxidation, so that can run in the 8 cycles or in the 8 phases or in the 8 times, and finally the fatty acids are metabolized. It can yield the lot of the ATP, and this fatty acid do not directly enters, okay, into the beta oxidation. So there is need to activations of the fatty acids or the separations of the fatty acids from the glycerol, that was known as the hydrolysis. And this hydrolysis process is occurs in the presence of the lipase and as well as the in the presence of the water. So additions of 3 molecule of water to the triglycerol, the fat hydrolysis is occurs, or the hydrolysis of triglyceride will be the occurs. So then the glycerol and free fatty acids, so they are separated. Then the free fatty acids are activated. Then so they are enter in the beta oxidation process. So this is the regarding the entry or starting of the beta oxidation process. So that was done with the help of the lipase enzyme. So that lipase enzyme, it was causes the hydrolysis of the triglycerol, then separate the glycerol as well as the fatty acids. So that fatty acids enter in the beta oxidations. So regarding the first stage, that is the activations of fatty acids, so that was mainly occurs in the cytosols. So in the cytosol, the fatty acids, it was activated into a acyl CoA. Okay, it was occurs in the cytosol, but the beta oxidation, it was occurs in the matrix of the mitochondria. But this fatty acid, it is not able to cross the membranes of the mitochondria. So there is need to activations of the fatty acids in the cytosol, then only, so that activated fatty acids in the form of the acyl CoA, so then only it can enter in the mitochondrial matrix, then it can go for the beta oxidations. And the activations of the fatty acids in the cytosol, so that was occurs in the presence of the enzyme thiokinase, or also in the presence of the or hydrolysis of the ATP. Okay, so like that the thiokinase enzyme, so it was responsible for the conversions of fatty acid, that is acyl chain, into a acyl CoA. So in the presence of the thiokinase enzyme and hydrolysis of ATP. So ATP molecule can be hydrolysis. That is 2 ATP molecule is required here for activations of the fatty acids. So then it can form the acyl aldenate. Then in the presence of coenzyme A, okay, so that was converted into a AMP, it can form the acyl CoA. And the enzyme required for activations of fatty acids is a thiokinase. It was also known as the acyl CoA synthetase. And regarding the coenzyme factor, COSH, ATP and magnesium, so these are required as the coenzyme as well as the cofactor. This is the stage 1 regarding the activations of fatty acids occurring in the cytosol, because so that fatty acid, whatever the fatty acid, so they are present in the cytosol, that cannot be enter into the mitochondrial matrix. And the beta oxidation, it was mainly occurs in the mitochondrial matrix. So there is need to activation. So that activations of fatty acids was done. So with the help of the thiokinase, ATP as well as the magnesium. So next one is the stage 2, that is the transports of fatty acyl CoA. So that was formed in the stage 1 due to utilizations of 2 ATP in the presence of the thiokinase enzyme. So that fatty acyl CoA transported from cytosol into the mitochondria. So with the help of the carotene. So here dear students, the carotene is a carrier protein. So that was helpful for transports of the acyl CoA from cytosol into the mitochondrial matrix. So here the two enzymes are involved. So those enzymes are helpful for the transports of the acyl CoA from cytosol into the mitochondrial matrix. So those enzymes are carnitine acyl transferase 1 and as well as carnitine acyl transferase 2 enzyme. Transferase 1 enzyme is found in the cytosol and transferase 2 enzyme it was found in the mitochondrial matrix. And these enzyme it do not cross the that is the mitochondrial membranes, and as well as acyl CoA directly it do not cross the that is the membranes of the mitochondria. Only with the help of the carotene, by the presence of this enzyme that is the carotene acyl CoA, it can convert acyl CoA into a acyl carotene and losing a coenzyme A. Okay, CoA SH. So this enzyme it convert acyl CoA, or it can separate the CoA from the acyl and join the carotene. Then it become the acyl carotene complex in the presence of this enzyme that is a carotene acyl transferase 1. So then only, so with the help of the translocase enzyme, so it can carries the transport the acyl carotene into a mitochondrial matrix. So where, so due to the presence of coenzyme A and as well as carotene acyl transferase 2, it can separate the carotene from the acetyl CoA. Then it can responsible for the formations of the acyl CoA, then the acyl CoA. So like that, from the cytosol it can enter into the mitochondrial matrix. Then it can go for the further stage of the beta oxidation. So that's why, so the catenin, so it was helpful for the transports of the acyl CoA from cytosol into the mitochondrial matrix. So that's why, so this transport mechanism is also known as the carotene shuttles. So next one, that is the stage 3, that is the also known as the proper oxidations that was occurs in the mitochondrial matrix. So dear students, so the beta oxidations that was occurs in the mitochondrial matrix, it mainly occurs in the 4 steps. That is the steps 1, it was known as the oxidation. So that oxidations can regulated by the FAAD linked dehydrogenase enzyme. And second step or second reaction, that was known as the hydrolysis, it was regulated by the hydratase enzyme. And the third steps, that was known as the oxidation, so that was regulated by the NAD linked or NAD dependent dehydrogenase enzyme. Then fourth step or fourth reaction, that was known as the thylated cleavage, so that was regulated by the thiolate. So like that, so these steps for each fatty acid oxidation it was run by a 8 times. Okay, finally it can form the acetyl CoA. So that acetyl CoA enter in the further in the TCA cycle. So like that, each fatty acid molecule it can go through the 8 cycle of the beta oxidations, and each cycle it can cleave the 2 molecule of the carbons and to form a acetyl CoA from the acyl CoA or from the fatty acids that was occurs in the mitochondrial matrix. So now we go for the steps 1 from the proper beta oxidation, so that was known as the oxidations. Okay, so this oxidation, it is the first reactions of the beta oxidation, so that was regulated by the acyl CoA dehydrogenase enzymes in the presence of the FAD. So this dehydrogenase enzyme, it is mainly depending upon the FAD or related with the FAD. It can oxidize the acyl CoA and to form a trans-enol CoA. then where the FAD is converted into FADH2, then FADH2 enter in the electrons transport chain where it can responsible for the formations of the 2 ATP. Okay, so that's why, so this reaction is also energy giving reaction. So that was energy is produced in the form of the FADH2. So that was oxidized during the acyl CoA into a trans-enal CoA in the presence of the acyl CoA dehydrogenase enzyme. This is the first step, so that of the proper beta oxidation. So next one, that is the step 2, that is the hydrolysis in the presence of the hydratase enzyme. So trans-enal CoA, so that was converted into beta hydroxy acyl CoA by additions of a water molecule, and this reaction hydrolysis was regulated by the enal CoA hydratase enzyme. So this hydratase enzyme responsible for to cause the hydrolysis of the trans-enal CoA and produces them beta hydroxy acyl CoA. This is the second reactions of the proper beta oxidation. So where the trans-enal CoA is converted into a beta hydroxy CoA in the presence of hydratase enzyme and additions of the water molecule. So next one is the, once again, that is the oxidation. So that was regulated by the NAD linked dehydrogenase enzyme or beta hydroxy CoA dehydrogenase enzyme. So this enzyme is caused the oxidations of the beta hydroxy acyl CoA into a beta keto acyl CoA. So where the NAD is converted into NADH plus H. So this is the third steps or third reactions of the proper beta oxidation, and further, so this NADH enter into the electron transport chain reaction, then it can yield the 3 ATP. So that's why the step 3 of the beta oxidation is also energy giving steps. So where the energy will be produced in the form of the NADH in the presence of the dehydrogenase enzyme. So last steps, that was known as the thyrolytic cleavage. So that was regulated by the thiolase enzyme. So in the presence of the coenzyme A. So where the, that is the beta keto acyl CoA was converted into a acyl CoA minus 2 carbon atom. Okay, in every step it can cleave the 2 carbon atom, it can loses the 2 carbon atom. So that's why acyl CoA minus 2 carbon atom, and it can produces the acetyl CoA, and this reaction was known as the thyrolytic cleavage. So that was regulated by the thiolase enzyme in the presence of the coenzyme A. So where the ketoacyl CoA is converted into acyl CoA minus 2 carbon atom and as well as the acetyl CoA, and this acetyl CoA further enter into the Krebs cycle and produces the carbon atom as well as the water molecule along with the ATP. And this acyl CoA, then it can enter in the step 1 regarding the oxidation in the presence of the FAD linked dehydrogenase. Then after that hydrolysis, then oxidation, then thyrolytic cleavage. So like that, so this cycle, once the fatty acids are acyl CoA entering the beta, that is the mitochondrial matrix, means it can undergo the 7 that is the 8 proper beta oxidation. Finally, in each step, it can yield the acetyl CoA molecule. In the last step, in the 8th reaction, the 2 molecule of acetyl CoA will be yielded. So those both 2 molecule, so that can enter in the Krebs cycle, then it can respond to the productions of the further ATP. So this is the various steps. So those are involved in the metabolism of the fatty acids. Suppose it should be the 18 carbon atom, and each cycle it can loses the 2 carbon atom, then it was responsible for the formations of the acyl, that is acetyl CoA. So next one, so regarding the ATP yield in the beta oxidation, so that is the fatty acids. It is the complex of 18 carbon atoms or oxidations of the 18 carbon atoms, and each oxidation process or in each beta oxidation, so two molecule of carbon atom, so that was cleaved or separated from the fatty acids. So like that 18 carbon atoms fatty acids, it can undergo the 8 cycles, and during 8 cycles it can yield the 9 acetyl CoA molecules, and those are enter in the TCA cycle yield the 9 GTP, and during 8 cycles it can produces the 8 NADH as well as 8 FADH2 molecule, and further these molecule, so they are enter in the electron transport chain reaction once again, so they yield the more ATP. So like that in each cycle or in the 8 cycle, the 8 molecule of NADH is produced during the beta oxidations. Okay, and those molecules, so they are enter in the TCA where they can produce the 27, and total 35 ATP are produced. That is the 35 NADH molecules produces, and one NADH molecule it can yield the 2.5 ATP. So that's why in each beta oxidations or complete fatty acid oxidation, it can yield the 87.5 ATP from the only NADH. So like that, due to productions of FADH2, 8 from the beta oxidation and 9 from the TCA cycle, and total 17 FADH2 molecule was produced, and each FADH2 molecule it can yield the 1.5 ATP. So like that we can got the total 25.5 ATP from the FADH2 when the beta oxidation was completed in the 8 cycles. Okay, from the GTP or from the entry of the acetyl CoA into T cycle, where the 9 GTP, that is the equal to the 9 ATP are produced. So like that the total ATP from complete oxidation of 18 carbon fatty acids, so that was yield the 122 ATP, that is 87.5 from NADH2 and 25.5 ATP from FADH2, 9 from the GTP. Okay, so like that total 122 ATP was yielded, that is the 18 carbon atom undergo the beta oxidation, and 2 ATP molecule, so they are required during the activations of the fatty acids in the cytosols. Okay, so due to that, that is the final yield of ATP from the fatty acids is a 120 net ATP. So like that the beta oxidation is the energy rich pathway, so that was yield approximately 120 ATP molecule from FADH to NADH as well as TCA cycle and in the GTP. Okay, so total 120 ATP yield. So like that, so the beta oxidation it was provide the lot of energy to our body for the regular biochemical reactions. So next one, so regarding the regulations of beta oxidations of fatty acids, so some they are enhance the accelerate the beta oxidation, and some molecule or enzymes or hormones, so they are inhibit the beta oxidations. So among them, so epinephrine, okay, it is also known as a noradrenaline, sorry, adrenalines. It is a sympathomimetic hormones, or it is also known as the neurotransmitter secreted from the adrenal gland or adrenal matrix. So this epinephrine it was stimulate the beta oxidation through a stimulations of the cyclic AMP dependent protein kinase. Okay, it can enhance the activity of cyclic AMP dependent protein kinase due to activations of protein kinase. The phosphorylation is occurs, or activations of HSL is occurs. HSL means hormone sensitive lipase. So this lipase, it can cause the hydrolysis of the triglyceride and more fatty acids are produced. So they enter in the mitochondria. Then they can undergo the beta oxidation. So like that, the epinephrine, it can accelerate the or enhance the beta oxidation by enhancing the formations of the lipase enzyme or activations of the lipase enzyme. So due to that, it can increases the beta oxidation. And hormones like insulin, so already know insulin is the pancreatic hormone secreted from the beta cells. Okay, so this insulin is always released when sugar level is high in the blood. Okay, when your sugar level is more than the normal in blood, on that time only the insulin was released from the beta cells of the pancreatic gland. And the insulin, it mainly inhibit the beta oxidation or it can decrease the beta oxidation. So that's why the insulin is known as the inhibitors for the beta oxidations. Okay, and here the insulin, it was inhibit the beta oxidation by causing the dephosphorylations of HSL, that is the hormone sensitive lipase. It can cause the inactivations of lipase enzyme and lack of in these activations of lipase enzyme, the fatty acid do not release from the triglyceride or the glycerol do not suffer from the fatty acids. So due to lack of the HSL enzyme or hormone sensitive lipase. So like that, insulin is considered as the inhibitor for the beta oxidation and epinephrine is considered for the accelerator or activator for the beta oxidation. So like that, so these are the two hormones. So they are mainly regulate the fatty acid metabolisms or beta oxidations. That is the epinephrine. So as well as the insulin.